Hey, welcome to our webinar today. My name's Ron Myers. I'm the Director of Editorial here at JoVE. Today we have a presentation from Dr. Liang. So she published with us in the past on a flow cytometry technique to measure different parameters in mitochondrial analysis. So she'll be presenting some new developments on the technique and kind of comparing and contrasting to related techniques. Um, I will say we're recording the webinar, so if you miss anything, we'll be sending all the attendees a recording after the fact. And we are also doing a Q&A at the end. So please add your questions if you have any down in the Q&A in the bottom, and we will address those towards the end. All right. Well, thanks again, everyone, for attending. And Christina, I will leave it to you.
Yeah. Okay. Then thank you for Roland and Smita. Thank you for organizing this webinar. It's been a lot of work, and thank you for all the attendants that I can't see you, but hopefully you will have questions that we can communicate afterwards. So now I will start sharing my screen. Can you see my screen now? I hope it works.
Yes, looks good.
Okay, great. Good afternoon or good morning, depending on where you are in the in Earth. My name is Christina Xiaoliang, and I'm a senior researcher and PI. I'm leading a mitochondrial stem cell group in Department of Clinical Medicine in University of Bergen, Norway. So today I'm gonna talk a little bit about one of the papers that we are published in JoVE paper. So first of all, I would like to go a little bit for the background of the mitochondria. I know most of you know the background. So mitochondria is a double membrane bound organelle in the cell plasma. It consists of an outer membrane here and also an inner membrane here. And inside the inner and outer membrane, there's intermembrane space. Mitochondria have their own mitochondrial DNA, which replicate and repair by themselves, which is very important to look at the mitochondrial functions as well. Mitochondria is very important since it produces the ATP for almost all different type of cells. Therefore, when there is damage or defect of mitochondria, it will impact the different type of cells, especially the high energy demanding cells, for example, the neurons. A lot of researchers have been found mitochondrial dysfunction is either a primary or secondary contributors or responses to many types of diseases, including many different neurodegenerative diseases, such as Parkinson's disease, Alzheimer's disease, Huntington disease, etc. Also, a lot of other diseases, for example, the cancer. But so far, there is still a challenge of measuring the mitochondrial functions since the mitochondria, they are dynamic and they are heterogeneous. So measuring the mitochondria currently have different methods. For example, the seahorse to measure mitochondrial respiratory chain. And also some people use the fluorescence microscope to look at the mitochondria and mirror the mitochondrial proteins, for example. But still, it is still limited snapshots of measuring the mitochondrial functions, which cannot be able to capture the full scope of the mitochondrial dynamics. Therefore, there is a crucial need for the different technological advancements that can more precisely and dynamically measure the mitochondrial functions and their proteins. And it is very important to establish the method to accurately measure the mitochondrial health within the neural cells, especially for the people who is looking at the neural-related diseases, such as neurodegenerative disease, Parkinson, Alzheimer, for example. Therefore, we have published in 2022 a novel flow cytometry-based method to measure multiple mitochondrial parameters in different cell types. In this paper, we try to describe using the human induced pluripotent stem cells and their derived neuron and glia cells. So here I will go very briefly about the paper that we published in 2021. As you can see, the name of the paper is Flow Cytometric Analysis of Multiple Mitochondrial Parameters in Human-Induced Pluripotent Stem Cells and their neural and glia derivatives. So within this protocol, we have described two powerful techniques. One, we described using the human pluripotent stem cells, which can be derived from human or the patient directly and being reprogrammed into the stem cells. From the stem cells, the iPS can be differentiated into many different types of cells. For example, the neurons and even in different stages of one cell type, for example, the neural stem cells and mature neurons, as well as glial cells. Of course, iPS can also be differentiated into many other type of cells, for example, the cardiomyocytes, if you're looking into the heart diseases, for example. The other powerful techniques that we are using in this protocol paper is we have used the flow cytometry to try to measure the mitochondrial functions and the mitochondrial related proteins. Give a little bit but short background about my flow cytometry. Flow cytometry, as you all know, I believe it is a laser-based technique that can be used to detect and analyze a single chemical or physical characteristics of a cell or the particles. So in our protocol, as I mentioned, that we have outlined two major parts. The first part lying on the left side of the presentation, which shows like how we differentiate iPS into through the neuroepithelial stage, that we can see them and start neuroinduction for 5 days. Then we can see the neural reset. Afterwards, we lift them into the suspension culture and we can see the neural spheres. So they maintain the neuron progenitor stage from now on. So afterwards, we can put them into the adherent cell culture and then expand them as a neural stem cells for around 20 passages. Then we can bank them and we can carry out we can just sort them out if we need it. From the neural stem cells, we can also differentiate them into glia cells, for example, astrocytes, and maintain them for a longer time to allow them to be a mature astrocytes. From the neural sphere, if we don't drop them into the monolayers as neural stem cells, we push them into the dopaminergic lineage and finally differentiate them into the DA neurons and allow them for longer culture to grow as a mature DA neuron. So the left part, we have described how we use iPS and differentiate them into different neuron and glia cells, for example, the astrocytes. After we have got these cell types, we try to measure them, use flow cytometer, for example, the Accurate that I showed here, to locate the mitochondria and measure the mitochondrial functions, for example, the mitochondrial membrane potential. And we can also measure the mitochondrial volumes and mitochondrial-related Rose production. At the same time, we can also use this protocol to measure the mitochondrial respiratory chain subunits, for example, the mitochondrial complex 1 subunit, mitochondrial complex 2 subunit, and mitochondrial complex 4 subunit, for example. We have also used TFAM as an indirect parameter to measure the mitochondrial DNA replication and try to count the mitochondrial DNA copy numbers. So here I show the bright field pictures under our culture. The first one, we show how iPS look like and different. After 5 days of the neural induction, we see the neural reset. Then we put them into the suspension cultures and then maintain like this neural spheres floating in the culture. So from neurosphere, if we want to make them as monolayers, as neural stem cells, we drop them into the adherent cultures. Then we can expand and maintain them as neural stem cells. Otherwise, we can use neurospheres and differentiate them directly to the DA neuron progenitors, then drop them into the monolayer cultures, and they differentiate into the DA neurons. As I mentioned, the neural stem cells can also be differentiated directly into the isocytes for over 1 month differentiation and 1 month maturation, which has been described in the protocol in detail. Once we got different cell types, we have to look at their lineage. So here I show the confocal microscope in different markers to look at first panel as iPSC. We have the iPSC show positively the SSEA4, OCT4, and isocytes they show positively the GFAP expression as 100 beta expression. DA neurons, we have used TH and tubulin beta 3 to locate the DA neuron and try to characterize them to show they are in the right lineage after differentiation. So for the next is the mitochondrial environment. In mitochondrial environment, we also have 2 different protocols. The first section is using the live cell staining, we can measure the mitochondrial map potential, volume, and mitochondrial rows. So here I show briefly how we managed for this protocol. So after we got different type of cells, we see the cells in either 6-Wells plate or different multiple Wells plate. The next we make the staining solution, which contains FCCP, TMRE to locate the mitochondrial map potential, and MTG, mitochondrial tracker green, to look at mitochondrial volume, as well as the mitosox red to look at mitochondrial ROS production. After we have made the solution, we load them into the cells and put them into the incubator for different time period. So in this method, we have put different dyes. For example, here we have used the 4 wells. The first wells we use we seed only the cells without any staining. Second well, we call them negative wells, where we have put the FCCP, TMRE, as well as MTG to block the mitochondrial map potential. The third well, we put the TMRE and MTG to do the double staining. The next well, we stained with mitosox red and MTG. So within this different design, we could be able to stain double staining and to look at mitochondrial different mitochondrial parameters at the same time. For example, the mitochondrial map potential is presented as TMRE as well as the mitochondrial volume. So after a certain time of incubation, we remove the solution, we wash the cells, and we detach the cells. Run the flow cytometry afterwards. So this is how we do for the live staining to look at the mitochondrial functions. And next section of the protocol, we have used the fixed cells to stain the mitochondrial complex subunit. Also look at mitochondrial volumes as well as TFAM to look at mitochondrial DNA replication. Oh, sorry. Wait. Okay, again, we seed the cells and we isolate cells into the as a cell suspension. Then first we fix them as a 1.6 PFA. After fixation, we use ice cold methanol to do the permeabilization. And afterwards, we wash the cells and block the cells with unspecific antigens. So after that, we use different antibodies to do the staining. Here in the protocol, we have used mitochondrial complex 1 subunit to look at mitochondrial complex 1. We have used the TFAM as a mitochondrial DNA replicate parameter, as well as TOM20 to look at mitochondrial volume. After washing the cells, again, we run the samples in the flow cytometer. So here I also showed briefly how we analyze on the flow cytometry once you have done the cell staining. So first you open the software and you set up different gate and speed. Then you start run the different samples from the negative cells from the beginning, make the negative and positive gating, then run all the samples and finally measure the fluorescence intensity. So in the data analysis and calculation from the flow cytometry, we can measure the median of fluorescence intensity in different staining wells. And we also can make calculations based on different laser, which represent different parameters. And then at the end, we can measure the total level, both total level and specific level for different parameters, which include we can measure the total level of mitochondrial man potential presented by the TMRE staining. We can also measure the mitochondrial rose production. We can also measure the mitochondrial volume, as well as the complex 1 subunit expression and TFAM expression. At the same time, since we are doing the double staining, we can also calculate the specific level by using for example, the TMRE or dividing with mitochondrial MTG, as well as specific rows by using the rose expression and dividing with MTG, etc. For example, the specific complex 1, specific TFAM dividing with mitochondrial volume TOM20. So by using this method, we can more specifically to look at how the mitochondrial function or mitochondrial proteins can be changed per mitochondria and per cell. So we also have used this protocol and try to differentiate the IPS carrying the POC mutation, which is a primary mitochondrial disease, into, for example, the DA neurons, and use both live cell staining protocol and fixed cell staining protocol to measure the mitochondrial functions and mitochondrial parameters. Here we can see by using this protocol, we didn't find big changes of the patient, POC patient DA compared to control. No changes for the total level of mitochondrial man potential. However, after the calculate with specific mitochondrial man potential, we can see the decrease of the patient DA. When we look at measure the total mitochondrial rose production, there's no significant change. However, after we calculate with specific level of mitochondrial rose production, we can see the overproduction of the metal rose. And also we have used a fixed cell protocol. We found let me move the bar. We can see both total level of different mitochondrial complex subunit, which complex 1, no significant change, complex 2, no significant change. However, after we calculate with a specific level, we can see a significant change of mitochondrial complex 1, which are expected. And however, there's no significant change for the complex 2, which is also, we also expect this result. When we look at the TFAM, both at total level and specific level, there's both have showed the significant change using this protocol. So at the end, I would like to draw attention of the advance of this protocol. We have shown this is a powerful tool for measuring the multiple mitochondrial parameters in a single cell level, where we have used human iPSC and their neuron and glial cells. This protocol also allows the researchers to measure the mitochondrial parameters as I mentioned, both at the total level and also the specific level, which we can calculate by per mitochondria using the flow cytometry. This protocol can also be applied to many type of cells. Here we have described basically the different type of neurons and glia cells. And I would like also to, I wouldn't go through this detail, but I would like to draw your attention that this protocol can also be used not only from 2D cell culture and also from into the 3D bringing organelle, which we have also published 2023. If you're interested, you can also take a look at this protocol paper. At the end, I would like to thank to the members who contributed to this paper, me, myself, and Lawrence Bindorf, who is a former leader. Now he retired, and PhD student Cecilia and Anbing, who contribute a lot for this, both the measurement of the protocol and writing for the paper. And without the grant funding support, this research and protocol paper cannot be finalized. At the end, I would like to thank you for all your attention, and I hope you will now have some questions that I may can answer you. Thank you.
All right, thank you, Christina, for the excellent presentation. And yeah, like you said, if anyone has any questions, please add those to the Q&A. And it looks like there's already a couple there already. Looks like there's about 5 of them.
Okay, let me see. Now I can see several questions. Let me try to answer all. Promise. So the first question is, what does the FCCP dimer? So FCCP is a mitochondrial uncoupler. So it actually, so you know mitochondria have the negative charge. So FCCP actually can go negatively to the charge. So it actually block the mitochondrial bound potential. That's why we have used FCCP to actually measure as a negative control. We do the TMRE staining. Okay, second question is, during the differentiation process, what kind of metrics did you use and did you notice difference between the different metrics? Thanks. Okay, so this is very practical and nice question. So during the differentiation, it's quite depending on which process you are mentioning about. We have many different process, first from iPS into neural stem cells, and then neural stem cells into the DNA neurons, which we do see a difference with by applying different metrics. For example, the PDL coating or laminin coating, which do differ a little bit for the differentiation efficiency and the maturation. Third one, some other protocols use methanol also as fixation agent. Did you test the same protocol with other? Yes. Yes, we have also tested using only the methanol by both fixation and permeabilization. By actually, it both works, and protein expression level is basically similar as when we measure in our cell culture. But the thing that we use PFA is we would like to, after fixation with PFA, we can actually store them a little bit longer time, other than we use methanol to start permeabilization and fixation. Because when we have many different type of cells, for example, the patient and control, we would like to collect all the samples and do the staining at the same day, same condition, and measure them at the same time. So it works that we have tried. but we prefer still to use PFA, but methanol can also be used both for fixation and polarization. You also can save one more step for washing. Yeah. What are the adaptation of for single cell analysis? Well, for example, single cell sequence. Yeah, I think there's many new technique that can be used. But depending on what you want to look at, you want to look at the genomic profile, or you want to look at the protein profile. Or in our protocol, we, especially for the living cells, we actually can mirror directly into the live cells for their mitochondrial functions, which I think is a very benefit for most of the research. Is it possible to mirror isolated mitochondria by flow cytometry? This is very good question. We have not tried before. Maybe we should try, but I know people can isolate mitochondria. But since all these markers, for example, the dye can go into the mitochondria depending on their mitochondrial potential, or it go band mitochondrial complex directly. So it's very specific. So we see there's no big need that we should first isolate mitochondria. But of course, there are some cells which have very kind of limited mitochondria. You might have to isolate the mitochondria first. Next question, is there any way we can mirror specific mitochondrial ATP without isolating the mitochondria? Mammalia cells may some staining or yes, we do have a method that we can measure the specific mitochondrial ATP, but not with use of luminescent AC that we can measure the total. Actually, it's not mitochondria related, mitochondria specific ATP, but it's a intercellular ATP whole ATP production. And then we can also measure the number of cells to look at the per cell, how intercellular ATP has been produced and generated. But if you want to look at more specifically to the mitochondria related ATP, then I would think I would recommend you should go for the seahorse. Would mitochondrial isolation and the flow cytometry analysis important for dynamic analysis in fission fusion? Yes, we also have tried to use different dye or staining to look at the mitochondria fusion and fission, and also the Western blot, depending on what you want to look at. There actually have some live cell staining that you can see and you can analyze dynamically the mitochondrial fission and fusion. Yes. Oh, yeah.
All right. Excellent. Well, thank you everyone for the great questions, and thank you everyone for attending today. So as I mentioned before, we will send out a recording of the webinar in the next few days, so you'll have that for your records. And I know there were a lot of questions. We didn't have time to answer all of them, so if you do have any additional questions, feel free to respond to the email, and we can help you at that time. And if anyone is interested in chatting with an editor to discuss publishing any related mitochondrial techniques, feel free to reach out to us as well. All right. Well, thank you, Christina, for the excellent presentation, and thank you to all the attendees.
Thank you all. Have a nice day. Bye.
Have a good day, everyone. Yep. Bye-bye.